Okay, welcome back, everyone. Okay.
Hello.
How's everybody doing today?
Good.
Okay. Well,
Doing good.
good. Glad to hear it. And so welcome back. And I do hope that you are okay and that your families are well at this time. So we are going to get deep into chapter 8 today. It's going to be quite an involved day with chapter 8. So I'll be getting started with that in just a moment. Let me just remind you, right, that if you're not looking at your syllabus, you want to be doing that to make sure that you're not surprised by anything. So just be aware that exam two is coming up in 3 weeks, right? It's less than a month away, okay? So just be aware of that and keep that on the back burner. Okay, Elena, I see your comment, I understand. Okay, all right, folks, do you have any questions for the good of the order before we get started with today's conversation? Okay, well, gang, let's do that, okay? We're going to get back into chapter 8. Let me share my screen with you here. Okay, and so at the end of last class, we were talking about DNA replication, okay? And I had started making a list of the starting lineup for you to put in your notes. Again, if you have your textbook available, if you have it up on your computer as a digital form, that would be great. We're going to be taking a look at table 8.1. We're going to be looking at figures 8.3, 8.5. Those are things you want to have access to get our conversation started. So it's been a little bit since we've been talking about DNA replication, because last class we talked about unit conversion. Okay, so what I want to do is just show you a second slightly different video on DNA replication to get our thinking caps warmed up again. Okay, so the version of DNA replication that I shared with you previously was a very nice cartoon of the process, right? And that video link is in your notes from about a week or so ago. I want to show you a video today that's a little bit more real world. And by real world, I mean, it's not going to be quite as cartoonish. It's going to kind of show you things more at a molecular level of detail about how DNA replication works and what it really kind of looks like in an actual cell. Okay, so let's watch this. I think it's about 3 minutes. And then we'll get back into talking about this process. Okay. So here we go. I have placed this link in your notes and I'll run this forward. Here we go.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Okay, folks. So, all right. So that was a very different visualization of the DNA replication process than I shared with you last time. And I have no doubt that probably this video was just a little bit more unclear than the last video. And that's okay, because you can learn this process by studying the first video that I shared with you. But I wanted you to have an opportunity to see what the molecular machinery inside the cell kind of really looks like in real time. Okay, I mean, it's kind of crazy, isn't it? It looks like you were transported to some foreign planet when you see what's all this stuff's happening inside cells. It's really a foreign environment. Okay, so do you have any questions from watching the video? Okay. So let me ask you a question from watching the video. Okay. From the video, how fast did the video indicate the helicase enzyme was spinning? Okay, thanks, Kit. I'll just go with Kit since he's the first one there. We know Kit sits on his keyboard, right? So it's okay. 10,000 RPMs, 10,000 revolutions per minute, right? 10,000. You think about that, folks. Think about any object, that you might think of in the environment spinning at 10,000 revolutions per minute. That's really fast. And that little machine's doing it inside the cell. Here's one more question. One more question. Do you think the helicase enzyme is consuming ATP to do its job? I mean, how's it spin? How does it get the energy to spin? Okay, and absolutely yes, right? Helicase is an ATP-dependent enzyme. And my point is that if you're going to spin any kind of molecule at 10,000 revolutions per minute, there's going to be a lot of energy that the cell is burning through just to make that molecule spin. Okay, so the broader point here is that even a process that we might be invisible to our eyes inside a bacterial cell or inside our own human cells, when you're replicating DNA, there is a tremendous amount of energy, ATP, being consumed just to move the machinery to enable DNA replication to take place. Okay. All right, so I hope that, gets your thought processes working there a little bit. Let me finish this starting lineup for us. We're almost done here, okay? So I define DNA helicase, that's the enzyme that unzips the DNA, the single-strand binding proteins. They stabilize the single-stranded sections of DNA and prevent them from folding back on top of themselves. DNA primase is the RNA polymerase enzyme that makes a fragment of RNA, which is needed to jumpstart the DNA synthesis. Let me mention something here, okay? And it's just a philosophical distinction, right? For those people that spend a lot of time thinking about biology, inevitably it comes, one of the questions that comes up is, well, okay, what form of nucleic acid, DNA or RNA, evolved on the planet first? It's kind of like a chicken or the egg argument you might have with someone over a pint of beer while you're sitting at the local pub. And so once again, is it DNA or RNA that was created first on the planet? The truth is we don't truly know. Nobody knows. But there are certain pieces of evidence which point to an answer, which suggest an answer. And so this piece right here that I'm highlighting says that RNA polymerase is an enzyme. DNA primase is an RNA polymerase enzyme that manufactures fragments of RNA needed to prime new DNA synthesis. So the observation that we need to use RNA to get DNA is somewhat suggestive that RNA was created on our planet before DNA. Is it definitive proof? No, and we probably will never have definitive proof, but it's one small piece of the puzzle which helps us conceive of how life might have been created on our planet, how the basic building blocks of life were first synthesized on the planet. Okay, because RNA is needed to make DNA, it suggests that RNA evolved on the planet before DNA. It suggests it. It doesn't prove it. Okay, next thing. DNA polymerase. DNA polymerase. Okay, DNA polymerase is the main enzyme here that's going to make most of the new DNA. Okay, so this is the enzyme that manufactures most of the new DNA. Truth be told, there are multiple versions of DNA polymerase present, even in a single bacterial cell like E. coli. We're not going to get into all the different versions. That's too much right now. But suffice it to say, DNA polymerase is the enzyme that makes the new DNA. Folks, do you have any questions about the starting lineup, the molecules involved in DNA replication? Okay, what I would like to do is walk through a schematic. We're going to draw out a schematic of DNA replication and think about the steps that have to occur to copy a piece of DNA. All right, so that's where we're going to go. Okay, so we're going to have a schematic of DNA replication. Okay. So let's say, and I would encourage you to draw your own notes at your house, right? I mean, follow along. If you have access to a couple different colored pens or pencils, that would be very helpful. Okay, so let's say we have a fragment of DNA here. And remember, my artistic abilities are not so great, but here we are, okay? We have a fragment of DNA here. You tell me in the chat window, what is the first thing that has to happen if we're going to copy the DNA? What is the first step? I didn't even spell that correctly. What's the first step? What do we got to do? Okay, thanks, Rachel and Kit. All right. And Ireland, thank you. We have to unzip the DNA. That's exactly right. We have to unzip it. Okay, so we have to unzip this beast. Okay, does anybody name? So just to move this along, sometimes I'll ask questions, sometimes I'll just give you the information, but you know that the name of the enzyme that's needed to unzip the DNA is DNA helicase, right? So you're going to have a helicase enzyme that does its job. And so I'll draw, a cartoony version of it, kind of like this. Okay, where we have the DNA that's partially separated there on the left, and we have this. Okay, which will be helicase. Okay, so you know, in terms of drawing numbers, that would be number 2 here. Okay, so here's a question for you. Here's a question for you. How does helicase know where to begin unzipping the DNA? Remember, folks, right now we're talking about bacterial DNA replication. So that means the DNA is part of a circular chromosome, okay? Remember, the bacterial DNA is a circular chromosome. Okay, anybody have a possible response to that? How does the helicase know where to begin unzipping the DNA? Okay, so let me just respond to a few comments in the chat, and I deeply appreciate all of your answers. You know, even when they're wrong, I appreciate your participation, okay? So Rachel, remember, we're not talking about human cells right now. We're talking about bacterial cells. They don't have 23 chromosomes. E. coli has 1 chromosome. So Chelsea, you answered, is there a certain order of bases that it reads? Yes, Chelsea, there is. Okay, and in fact, take a look at Chelsea's comment. Is there a certain order of bases that it reads? Yes. The specific order of bases that is read has a special name. What is the name? Oh, Chelsea, bless your heart. It's not the Tada box, but I can tell you've been studying, or maybe you've had this in a different class. Okay, so it's not the Tada box. Chelsea, the Tada box is part of a promoter. Okay, but we're not there right now. Okay, we're not talking about that process. Anybody else? It's something that I would hope you picked out of your reading of the textbook. Let me help you. Take a look, figure 8.6, figure 8.6. The answer is on figure 8.6. What's the answer, folks? Figure 8.6. Come on, it's there, figure 8.6. It's not the promoter, no. It's not the replication fork, no. Thank you, Chelsea. It's the origin of replication. That's exactly right, the origin of replication. Okay. Okay, so you have to ask yourself, what is the origin of replication? What is this beast? Okay, let's define it. The origin of replication is a DNA word. It is a DNA word. Isn't that kind of a peculiar way of expressing it? This is a DNA word that is the binding target for the helicase enzyme. The DNA word that is the binding target for a helicase enzyme. Okay, and let's see here. I really want to get another page in here, so oh, technology is not being my friend today. Okay, but I'm going to keep going with this at the moment. All right, so let's talk about what this definition of the origin of replication means. Okay, and so all right, DNA word. You should think of DNA as a nucleic acid alphabet. Okay, so let me ask you, please just type your answers into the chat window. How many letters are available to us in the English alphabet? How many letters in the English alphabet? Okay, 26, right? 26 letters. And in the DNA alphabet, there are 4 letters, are there not? Thank you, good participation. And think about it, in the English language, if I write this sequence of letters, that sequence of letters on your screen, B-O-A-T, is a symbolic representation of a physical object that you can think of in your mind as a boat. B-O-A-T is a word, which is a symbolic representation of a physical object, a boat. If I use these same letters in different order, T-A-B-O, that is not a boat, right? It's not a boat because the precise sequence of the letters matters. The sequence of the letters matters because if I have B-O-A-T, it's boat. If I have T-A-B-O, it's gibberish. Okay, so in the same way, when you have a stretch of DNA like the origin of replication, and I honestly don't have the origin of replication sequence memorized, okay, that's too much. But for example, this might be an example of a DNA word that could be the origin of replication. Okay, that could be the origin of replication word. That could be that. It's kind of like hippopotamus, right? It's a big, long word, but that could be, this could be the DNA origin of replication. All right, folks, let me just ask you in the chat window. Is this making sense? How about everybody? Are you okay? Is there any questions you'd like to ask? Okay.
I have a quick question.
Okay.
Is the origin of replication the sequence of A, T, C, and G the same across DNA of all organisms, or is it
No.
different?
Good question, Taylor. No, it is not. I will say within certain subdivisions of taxonomy, the system of how we organize the relatedness, the genetic relatedness of organisms, you will find clusters and groups of similar bacteria like E. coli, Vibrio cholera. They would be expected to have very, very similar, if not identical, origins of replications. However, if you were to identify an origin of replication between an archaeal cell and a bacterial cell, they would be quite a bit different. So there is no one common origin of replication sequence across all species of life on the planet. There is variation. Okay. Other questions? Okay, so don't forget, folks, this origin of replication that we're writing out as TACGG the whole way out, don't forget that these letters represent nucleotides, right? They represent nucleotides. And don't forget that nucleotides are three-dimensional molecules. They're three-dimensional objects. Just like a real boat is a three-dimensional object, so are the nucleotides of the DNA that make up the origin of replication DNA word. It is a three-dimensional object. So what I'm telling you is when a three-dimensional protein like helicase binds to a three-dimensional stretch of nucleic acids, it's kind of like solving a 3D puzzle, okay? A 3D puzzle, okay? Question in the chat window. Have you ever built a 3D puzzle? Okay, you know what I'm talking about here. Okay. All right, so let's just do a quick Google search. Okay. You all know what a jigsaw puzzle is, right? A jigsaw puzzle, you put the puzzle together on the table, right? And they lock in based on the overlapping shapes of the pieces, right? Well, now take that jigsaw puzzle, and rather than having it two-dimensional, make it three-dimensional. A three-dimensional jigsaw puzzle. Let's just do a quick Amazon search here. Okay, they actually sell these kinds of things, and depending on how geeky you are, right? I mean, you might like to make one of these things, right? Here's a Disney castle that you can build in 3 dimensions, right? And that's what I'm trying to get you to see, that even inside the cell, these tiny little molecules are 3-dimensional. So the machinery of the cell is 3-dimensional, and the interaction of these enzymes with DNA is working in a 3-dimensional world, even though it's a very tiny world. Okay. All right. So my wife and I have done a couple of these. They are definitely harder than a 2D puzzle. Okay. Any questions? All right. So I lost my number 2 above, so we're just going to go forward here. Okay, so number 2, now we have to get to number 3. What would you say is the next step in the chat window? What's the next step that needs to happen for DNA synthesis to occur? Okay, Kit is suggesting that we have to prime the strands. That's true, and I'm going to run with this because this is a very deep topic that we could waste a lot of time on. But the idea here is that I need to prime the machinery so that replication can occur. And so that is going to take place with the activity of DNA primase. In other words, DNA primase is going to synthesize small pieces of RNA. Okay, so I'm not much of an artist, but we'll do it this way. Okay. All right. And so I will make the snippets of RNA. I'll make them little pieces of orange here. Okay, little pieces of orange. So this is RNA. This is an RNA primer. And that's going to be made through the activity of the DNA primase enzyme, which, you know, I'm not really going to draw everything up all the time because I'm a poor artist and it's going to get very crowded up here. Okay. Along the way, however, we do need to start paying attention to the directionality. Remember that conversation we had about the directionality of the strands of DNA, that they run anti-parallel to each other? We need to start paying attention to that. Okay. So, all right. If I make the directionality of this strand of RNA 5' here and 3' here, my question is this, right where I'm putting a question mark. Elena, bear with me here. I think we'll get you through this, okay? Hold on. So my question to you in the chat window is this. considering the directionality of the RNA, what would the question mark on the DNA be? Would it be 3' or 5'? Okay, good. Yes, it would be 3'. That's exactly right. Okay, so this strand of DNA, which is running anti-parallel to the RNA, is actually 3' here and 5' here. Okay, and the bottom strand of DNA is going to be 5' here and 3' here, right? Because it has to be anti-parallel to the other DNA strand. Okay, so that means that the other RNA primer, right, is going to be 3' here and 5' there. Okay. All right. So DNA primase would add the primer. DNA primase adds the primer. Okay. Forgive me for asking, but I really don't want to lose you. So are you okay right now? Is this making sense? Okay, it's one of those things where you have to walk through it, and you probably have to watch the video like 5 times and think about what's happening to really kind of get things here, right? Really kind of get things here. Truth be told, I actually forgot a little piece of information, and I'll just put it in here for you. Okay. So that's my bad. When helicase unzipped the helix over here, as we went from 1 to 2, it would have been the case that we would have had some SSB molecules, right? Some SSB, single-stranded binding proteins, would have stabilized those stretches, and that would have occurred on both strands there. We would have had single-strand binding proteins attached to the nucleic acid and stabilize it. Okay, because if you have, if you have a piece of DNA that starts out as a helix, and then you unzip part of it, the ends that are unzipped can actually fall back on top of themselves like this. Oh, come on here. They can do this. Okay, they can do that. And if that happens, this would block DNA replication. Kit, you're asking, could that result in a mutation? Yeah, it would be possible that could promote the creation of a mutation. That is possible. Okay. All right. So step 1, we unzip. Step 2, we add some single-strand binding proteins. Step 3, we create little pieces of RNA primers to prime the reaction. I don't know what your experience in life is, but let me ask you this. Have you ever mowed the lawn? Okay. Hopefully you've all mowed the yard, right? Now, obviously, there are many different kinds of lawnmowers out there, and you might be fortunate enough to have a really big, sophisticated riding mower, and it's all nice and nice. But some of the older style mowers, like the walk-behind mowers, they have tinier engines, and at least in some of them, and in one that I currently own, you actually have a tiny little rubber bulb, at the engine where you have to push the bulb in to inject gasoline into the carburetor of the motor for the mower. And that's called the primer. You push the little rubber bulb called the primer, and that pushes gasoline into the carburetor. Okay, that's kind of like what we're doing here. We're not pushing gasoline into the carburetor, but we're putting a piece of RNA into the DNA structure for the purposes of DNA synthesis. Okay. All right, so I'm going to continue down here. I'm sorry, I wish I had a giant screen that I could put everything on, but I'm going to continue my conversation off of number 3 at this moment of time. Okay, so I'm going to come down here and we'll look at this. And we have DNA helicase. And DNA helicase is moving in this direction to unzip the helix. We have some single-stranded binding proteins that are on the single-strand fragments of DNA. They hop on and off as other enzymes come across the template. I'm not going to redraw them at this moment. I have fragments of RNA, right? Like this one here, a fragment of RNA. And I have the directionality of the molecule, which I want to honor and stay faithful to. So the blue molecule, the blue strand is 3', 5'. And so that means the orange strand can be 5' at the top and 3' at the bottom. And that's going to happen on both strands, okay? So what you're going to have next is DNA polymerase is going to come in here. Okay, DNA polymerase is going to sit down here. And I am really not much of an artist. Okay, so let's see, let's use maybe a light green color here. Okay, so you're going to have DNA polymerase sit down in this pocket of space, which is the replication fork, and it's going to begin. DNA polymerase is a big enzyme, and it has multiple arms. It has multiple arms. Okay, folks, do you, question in the chat window. Do you know who Dr. Octopus is in the Spider-Man comics? Okay. If you don't, go ahead and look that up. Google Dr. Octopus and Spider-Man, okay? At any rate, he's this villain in the Spider-Man comics that has multiple metallic tentacles, and you can independently move the tentacles around to grab objects. Well, that's DNA polymerase. DNA polymerase is like an octopus. It's like an octopus. It has multiple arms. One of the arms of DNA polymerase comes up here in this area and starts making new DNA in the same direction as the helicase is moving. And another arm comes down here and does sort of the same thing. Okay, so you can see how ugly that drawing is because your instructor is a poor artist. But DNA polymerase has multiple arms. Okay. And so what that produces is a form of replication which you can see similar to figure 8.5. Figure 8.5 in your textbook. Okay. Let me ask you in figure 8.5. In figure 8.5, how many DNA polymerase enzymes are shown? How many are shown? in figure 8.5, how many DNA polymerase molecules are shown? Okay, there's multiple ones, right? There's multiple ones, okay? Here's the deal. The DNA polymerase enzymes that are drawn in your book are not quite drawn accurately. The real deal is that DNA polymerase is a single molecule with multiple arms. So you can think of DNA polymerase as an octopus with 3 tentacles in figure 8.5, okay? That's the basic idea here. All right, so long story short, there are 2 forms of DNA strand replication. We are going to have leading strand replication, and we will have lagging strand replication. Okay, so can anybody tell me, okay, so let me finish the drawing here. On step 4, DNA polymerase at the top is moving towards the helicase, and DNA polymerase on the bottom would be momentarily moving away from the helicase. Can anybody tell me, is it the top strand of drawing number 4 or the bottom strand of drawing number 4, which is illustrating leading strand replication? Okay, it looks like most of you are picking this up. Good. I'm very happy about that. Okay, it is the top strand. Okay, so leading strand replication is replication where the arm of DNA polymerase is moving naturally in the same direction that the helicase enzyme is unzipping. Okay, for leading strand replication, DNA polymerase moves in the same direction as, as the helicase. Okay, lagging strand replication, the DNA polymerase arm, if you will, is extending away from the natural motion of the helicase. Okay, so let's see. I should bring up that video that I showed you last time and review part of it with you. Okay, so let me get my notes here. All right, here we go. Let's watch part of this video again.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the 2 parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, sorry for the just abysmal background music. I don't know why they put it there, but okay, come on. Okay, all right, folks. Tell me what's bothering you right now about this topic. Tell me what you need support on to understand this stuff. Okay. Is there anything you'd like to ask about? All right, I get a little nervous when there's so much quietness. Does that mean?
I actually had one question.
Okay.
In the drawings that we did, do the SSBs, are they like in between the RNA primer and the helicase, or are they after the primer?
Okay. Good response. Good question. They can be at both locations, but they're only on the single-stranded nucleic acid long enough to stabilize it. So when DNA polymerase sits down on the RNA primer and starts to move towards an area where there would be a single-strand binding protein, the helicase bumps the single-strand binding protein off.
Okay, so it doesn't stay. Okay.
Right, it doesn't stay. Okay. However, if there remains an area of single-stranded DNA that's exposed and there's nothing moving along that stretch, then the single-strand binding protein can reattach.
Okay. Thank you.
So Elena, you're asking, the video we just reviewed made it more visual for me. Why did it curve? Please help me understand your question.
Yeah, at the end of the, like towards the end, it just showed kind of it curved, and I know you talked about how the octopus kind of like all over the place. Is that just kind of the process, like it just all over?
When you say curve, what part are you
Right there,
talking about?
when you see at the end, kind of, you see how it took like that turn?
This turn here?
Yeah.
Yes.
Like, is that just part of the process? It's just all over the place.
Well, it is part of the process, and it may seem like it's all over the place, but there is a logic to it. Remember this, that the two strands of DNA, and I know this is getting very busy here, the two strands of DNA are running in opposite directions. So this is 3', 5', 3', 5'. So DNA polymerase on the top strand can move this way naturally. DNA polymerase on the bottom strand cannot naturally move in the same direction because its template is in the opposite direction. So what the enzyme will do is take the DNA template and form a little loop with it, which is what you're seeing in the video here. That's that little loop. It will form a little loop so that the enzyme can actually momentarily move in the same direction to copy the DNA. That is a characteristic, Elena, of lagging strand DNA replication. Maybe we should put that into your notes. Okay, lagging strand replication always involves looping
Okay, thank you. That makes more sense.
the template DNA strand. Okay. Folks, you know, it's my job to push you a little bit on this, but you know, don't get too worried. I'm honestly not going to have a giant essay on the exam, on next exam. It says, you know, explain all of this with a giant cartoon. I'm not going to do that to you. Okay, but you should at least review the videos, review the notes, know the names of the primary machinery involved in the process, and know some of the vocab words like origin of replication, that kind of thing. Okay. Other questions? Okay, so I will point out that the lagging strand, yes, Kid, I see your comment. I will point out that the lagging strand is made in a start-stop motion, like the video was saying, sort of a stuttering motion. Okay, and it has to do with the looping mechanism of the template strand. That start-stop motion will create tiny little snippets of DNA, which are called Okazaki fragments. Those fragments were named after Dr. Okazaki, who originally discovered them during his research. And so Kit has a question. So DNA ligase just bonds the Okazaki fragments together? Basically, yes, Kit, it does. Okay. All right. So we need to wrap this up so we can move on to the next topic. Okay. But I will point to figure 8.6, right? Figure 8.6 shows the process of DNA replication taking place along the circular chromosome. And what I would highlight to you is that if you're looking at figure 8.6, look at panel B of figure 8.6. So if I start with the circle of DNA where you see the phrase origin of replication, and I start counting with that circle, number one, I go below it, number two, go below that, number three, go below that, number four. The fourth picture of panel B has effectively what looks like magician's rings that are interlinked. Magician's rings, the metal rings that get linked during a magic act. That's what you're going to have here taking place when you copy a circular chromosome. And so suffice it to say that we'll wrap this up quickly here. Okay, number 5, stopping DNA replication. Okay, stopping DNA replication happens when you reach what is known as a terminating region along the chromosome. So this whole process got started with a DNA word called the origin of replication, and stopping it's going to have a different DNA word, which is called the termination sequence. It's here that you're going to have the magician's rings. Okay. So you notice in figure 8.6, panel B, we go from the fourth group of rings, which I was trying to show you as magician's rings, and we go one more below, and all of a sudden the magician has unlink the rings somehow. Okay, so the unlinking of the rings, the unlinking of the replicated DNA is due to a special enzymatic machine that comes in and literally cuts the links apart, cuts the links, holds on to them, physically separates them through distance, and then reseals them. That enzyme is called DNA gyrase. Okay, DNA gyrase. Okay, so DNA gyrase is an enzyme listed in table 8.1. Okay, and it says DNA gyrase relaxes the supercoiling ahead of the replication fork. Don't worry about what supercoiling is. I can tell you, but I'm not going to test you on it. Okay. DNA gyrase is the enzyme that's going to unlink the magician's ring. It's going to unlink the two copies of DNA at this point, and now we have two circular chromosomes. Okay. All right. So, folks, if you're still feeling fuzzy about this, first off, you're not alone. It's not like people figured this out in a Saturday afternoon, right? It's like people figured this out after about 50 years of research, okay? So we're taking a lot of complicated stuff and trying to give you the basic idea here. Would you like to ask any questions about DNA replication? Are you sure? I'm going to have to fix this document at some point because it's just not what I want it to be. I'm going to start a new note-taking document here. Okay. Let me know if you have questions. Yes, Aubriana, I see your comment in the chat window. Okay. The book says gyrase relax the strands. That's right, it does. Okay, and without going into too much additional detail, let me just make a cartoon here. Okay. When you talk about the structure of DNA, which is getting way beyond what we would talk about in this course, okay, typically you have a helix, right? So here's the helix. This is the natural helix of DNA. So, Aubriana, when the book starts talking about gyrase relaxing the strands, what that means is this. If what happens in their actual structure of DNA is that DNA starts to actually fold over on top of itself, where it crosses itself is called a supercoil. And if you want to replicate DNA, you have to remove supercoils so the machinery of the DNA polymerase, all the machinery at the replication fork can do its job. And so DNA gyrase will remove the crossings temporarily so that the enzymes can actually copy the DNA. Other questions? Did that make sense, Aubriana? This is not a trivial topic. I mean, there's a lot of research that still continues on just figuring out the nuances of DNA replication. Okay, please let me know if I can help you, and I want to encourage you to review notes, look at the videos like 20 times and really think about what's happening here. That will be that will be a good place to be at when you're taking your next exam, okay? All right, so we're going to move on from DNA replication. And I will remind you that DNA replication is like watching, excuse me, I just made a mistake. DNA replication is like copying the movie, We're going to move on from copying the movie to actually watching the movie. Okay, so we're moving on from DNA replication, which is copying the movie, to gene expression, which is watching the movie. To watch the movie, doesn't your DVD player, your Blu-ray player, I know for the super younger generations, it's like, what is that? I just stream everything. But the Blu-ray player has to literally decode the information on the physical disc. That's watching the movie. That's gene expression. So we need to move to that. Okay, so gene expression, part 1, which is transcription. Now, I'm not going to spend as much time on these processes as I did with DNA replication, right? I spent well over an hour with DNA replication between today and last class that we talked about it. We don't have the luxury of that amount of time, okay? But I do want to give you the basic ideas of DNA, of transcription, okay? So transcription is the name of the process that you make the RNA, okay? Name of the process used to make RNA. The primary enzyme that does the work is RNA polymerase, okay? Transcription as a process is graphically illustrated for you in your textbook, figure 8.7. Figure 8.7. And so that's, that's a really good starting point for you. Let me play one of the videos that I have queued up for you to at least give you some visualization on how the process of transcription works. I'm not going to draw a cartoon schematic with that one, but I will put a couple notes into your file here so that you have some background when you study. Okay, so let's see here. And all these videos I'm showing you, I have provided links in the chapter, in the notes that I'm giving you, okay? So you'll get them later today. All right, so let's watch this video on transcription. Transcription is the process of making RNA. Here we go.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
Okay. All right. So, fairly complicated as well. But if you get the rough idea from the video and know some of the basic vocab that's shared with you in this chapter, you're going to be okay. All right. Okay, Elena, thank you. All right. Do you have any questions from having watched the video one time? Chelsea asks, what causes DNA to rewind after it leaves the RNA polymerase? There is no molecule that causes the DNA to rewind, Chelsea. The two strands of DNA have a natural electrical attraction to each other through the nucleotides that are on opposite strands. So if there's nothing forcing them to stay apart and they're close together, they will naturally reseal, kind of like rubber on a tire that kind of reseals over a small puncture hole. Okay. Other questions? Okay, so let me highlight some features of transcription that hopefully are going to come out of your study time, out of the textbook, and out of the videos that you're going to be watching here. Okay, so transcription is the name of the process used to make RNA. And that means all forms of RNA, not just messenger RNA. Okay, so if the phrase messenger RNA is strange to you, just give me a couple minutes and I'll clear that up for you. Okay, but whenever a cell makes RNA, whether it's transfer RNA, messenger RNA, ribosomal RNA, other forms of RNA that are not even in your textbook, that process of making that type of RNA is transcription. Okay, the primary enzyme that does the work is RNA polymerase. Here's a question for you in the chat window. Okay, where you are going to have trouble as a student, at least what my experience tells me, is that you're going to understand DNA replication, and you're going to understand transcription, and you're going to understand translation. But when you try to change between one topic to another, some of the molecules that are associated with one process, you're probably going to get them confused with an alternative incorrect process in your mind. I've seen that happen 1,000 times. So please be careful here, okay? Okay, during DNA replication, what is the name of the enzyme that unzips the DNA? Okay, thank you, folks. It's helicase. Well, guess what? During transcription, we have to unzip the DNA too, right? We have to unzip the DNA to make the RNA. But in transcription, there is no DNA helicase enzyme being used. So my point here is that in transcription, we unzip the helix differently. And you know what? It's actually pretty simple, because RNA polymerase is the enzyme that not only makes the RNA, it unzips the DNA, okay? So RNA polymerase can do two things. Okay, it will unzip the DNA, and I'll put in parentheses, this was done by DNA helicase during DNA replication, and it can manufacture RNA. Okay, so RNA polymerase is the big honcho here. There are other enzymes involved, other proteins, other machinery. We are presenting a simplified perspective to you, but that's okay right now. We're just trying to get you into the topic. Okay, so RNA polymerase can do two things. It can unzip the DNA and it can manufacture the RNA. What else? Okay, number 4 here. So just type into the chat window, folks, how did DNA helicase know where to start unzipping the DNA? How did it know that? Right, the origin of replication for DNA replication. In transcription, we have to know where to start unzipping the DNA for transcription. But the signal is not the origin of replication, because we're not doing a copying process. We are expressing a gene. So RNA polymerase knows where to begin unzipping the DNA by recognizing that term that some of you have been throwing out called the promoter. Okay, so let's define that. A promoter is a DNA word that acts as a landing pad for RNA polymerase to begin unzipping the DNA. Okay. In many practical ways, if you can identify the promoter in a stretch of DNA, you have identified the start of a gene. Basically, the promoter is the beginning of a gene. Okay, so RNA polymerase is recognizing this sequence of DNA nucleotides, which we are calling a promoter, attaching to it and starting the work of unzipping the DNA so it can start to make fragments of RNA from the DNA template. Okay. All right. Now we have to deal with, well, how does RNA polymerase know where to stop? You know, where does the gene end? And you can see for yourself on figure 8.7 that each gene has a sequence that's called the terminator. Okay, so a couple things. First off, every gene has a little Arnold Schwarzenegger sitting at the end, and he says stall. Okay, every gene has a terminator sequence. Terminator sequence sounds a lot like the termination sequence during DNA replication. They are different sequences, but they have basically the same function, which is to stop the process. Okay, so RNA polymerase will stop making RNA when it scans across a DNA word called the terminator. So here in the context of transcription, we have defined terminator, we have defined promoter, right? The words promoter and terminator are pieces of language that are specific to transcription. If you applied them in the context of DNA replication, it would be nonsense because these words are not relevant for DNA replication. These are words that are relevant for transcription. Okay. Folks, do you have any questions you'd like to ask? All right. I know I'm already over time. My apologies. We do have lab today. I'm going to continue lecturing in chapter 8 so we don't get behind in our curriculum. So probably 30 to 45 more minutes worth of lecture, and then we'll start introducing this upcoming lab activity that you have, okay? So let me stop right here. Okay. So before I leave the topic of transcription, it might be useful for you to think about how similar DNA replication and transcription really are, right? Both processes involve unzipping the DNA. Both processes involve sort of a start signal. For DNA replication, that was the origin of replication. For transcription, that's the promoter. Both processes involve a termination process. Okay, for DNA replication, that's the termination sequence. For transcription, that's the terminator. Okay, so there is actually a lot of similarity between these processes. Obviously, DNA replication, you're making a copy of the DNA. Transcription, you're taking the DNA and changing the information into a version of RNA. That's what makes it gene expression. Okay. Is there anything you'd like to ask at this time? Okay. So I'm going to move on to the second part of gene expression, which is translation. Okay, translation. All right. So translation has a big, long figure in your textbook, figure 8.9. Gene expression, part 2. Okay. Translation. This is figure 8.9. Okay. Okay. So let me give you some talking points here. Okay. In order for translation, which is the production of a polypeptide, right? The production of a protein. Okay, so we'll say that first, right? Translation is the process of making a polypeptide. Okay, so here's a question. Technically speaking, there is a difference between polypeptide and protein. What is that difference? Does anybody know from any other coursework you've had? Technically speaking, there is a difference between a polypeptide and a protein. Do you know what the difference is? Okay. Kit, thanks for contributing. And in this case, you're not correct. You've been doing a great job. Okay. Not so much the size difference. Chelsea, you're saying how they are wrapped. I'm not sure what you mean by that. What do you mean by that, Chelsea? Well, okay, we'll continue here. Chelsea, okay, so Kit, you talk about structure. That's getting to it, okay? Folks, a polypeptide does not have a defined shape, but a protein does, okay? A polypeptide does not have a defined shape, but a protein does. As an analogy, okay, a polypeptide would be like your shoestring if it's out of your shoe and you're just holding the shoestring at both ends between your hand. It's just kind of a limp thread of fabric. It's just a shoestring, okay? So the polypeptide is like an unfolded shoestring, and a protein is if you take your shoestring and you make a bow out of it to tie your shoe. Okay, the protein has a defined shape. In my analogy, creating a bow, okay? It's like tying your shoe. Okay. All right. Chelsea, your comment, there's absolutely no reason to apologize, okay? Proteins can have 4 levels of structure: primary, secondary, tertiary, and quaternary, right? And if it has quaternary structure, as you're writing in the chat window, that means there are multiple polypeptides that are interacting with each other and folding into a defined geometry, a defined shape. That is absolutely correct. Okay. All right. So, all right. In order for translation to work correctly, there's got to be a bunch of machinery. Okay, so that machinery is going to involve the ribosome, right? So translation's main machine is the ribosome. That's what you kind of graphically see simulated on figure 8.9 is this complex called the ribosome, right? Remember, ribosomes are made of many proteins, okay? For example, an E. coli ribosome has about 55 ribosomal proteins. And different forms of a type of RNA called ribosomal RNA, and 3 different forms of ribosomal RNA, okay? Translation's main machine is the ribosome, okay? Now, for translation to take place, there's going to be 3 critical forms of RNA that are involved in the translation process. And those forms of RNA are all manufactured by what process? Type your answer into the chat window. All forms of RNA are manufactured using what process? How about everybody? Okay. Yeah, no, Rachel, no, it's transcription, right? RNA is made using transcription, not translation, okay? All right, so 3 forms of RNA made from transcription are needed to support translation, right? Isn't it kind of horrible that transcription and translation both start with the letter T, and if you're not careful, they can sometimes sound similar, and we get them confused, right? Just be careful. So 3 forms of RNA made from transcription, and that is not a typo. That's actually correct, okay? 3 forms of RNA made from transcription are needed to support the translation process that ribosomes execute. Here's the 3 forms of RNA. We have messenger RNA, so-called mRNA. The messenger RNA is going to be the primary template of the ribosome. The primary template of the ribosome. The ribosome is going to sit on the messenger RNA and start decoding it. Next, we have transfer RNA, tRNA. This is, if you've had any biology, this is the molecule that brings amino acids to the ribosome, because you know polypeptides are made by linking amino acids together into a chain, right? So transfer RNA is the molecule that transfers an amino acid to the machinery of the ribosome so it has something to work with, okay? This is responsible for bringing amino acids to the ribosome. Think of transfer RNA as a Federal Express delivery guy, and his job, her job, is to bring amino acids to the ribosome. Next, ribosomal RNA. A ribosomal RNA, this piece of RNA is a structural component of the ribosome. If you were to break apart a ribosome and look what's inside, you would find ribosomal RNA. So it's a structural component of the ribosome, and it might be of interest to you that the ribosomal RNA in the ribosome is also responsible for joining the amino acids together. It's the piece of the ribosome that's responsible for catalyzing the formation of peptide bonds. Okay, so this is a structural piece of the ribosome and is responsible for linking amino acids together. Okay. So do you remember earlier in our conversation today, I told you that one of the reasons we think RNA evolved before DNA is because you need a piece of RNA to begin synthesizing DNA. That's one piece of evidence of why we think RNA may have formed before DNA. Here's a second piece of evidence. Okay, so let me ask this question in the chat window. Here's a question in the chat window. Here you go. What category of organic macromolecule do you associate with the term enzyme? Right, so our options are carbohydrates, lipids, proteins, nucleic acid. Okay, thank you for your answers, folks. It is protein, right? Enzymes are thought to be protein. And you know what? 99.5% of them are. If you think about an enzyme, it is almost always a protein. But there are some exceptions that have been discovered by science over the decades of work. And that exception are catalytic pieces of RNA, which are called ribozymes. Okay. Ribosomal RNA is an example of a ribozyme, which is a catalytic piece of RNA, similar to the enzymes made of protein. Okay. And so if you're curious, you can Google ribozyme and you'll get all kinds of stuff that you probably never want to read. Okay. But the truth of the matter is that the part of the ribosome that's responsible for linking amino acids together is not a protein-based enzyme like we would think. It is an RNA, a ribosomal RNA enzyme called a ribozyme. Okay. So for this second observation here, when you think about the evolution of nucleic acid on planet Earth, you got DNA, you got RNA. We have never observed, I mean, humanity, scientists, human beings, we have never observed DNA as having any enzymatic activity. As far as we know, DNA cannot act like an enzyme, but RNA can. And the biggest example is the ribosome. Because RNA, at least in some cases, can have enzymatic activity, this is another small piece of evidence which is suggesting that RNA evolved on the planet before DNA. Okay? It's kind of one of those things that's philosophically interesting to consider, but knowing it is never going to get you a job. Okay? So it is what it is. So let me show you a translation video at this time, then we can wrap up our note taking here, at least on this topic. Do you have any questions you'd like to ask? Okay, all right. I hope that doesn't mean you're passed out at home. Okay, here we go. Let me bring this up. Okay, excuse me. Okay, here we go. How translation works. This video is linked into your class notes that I'll send out later. And here we go.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so fairly, quick 2-minute video, but it does a nice job of sharing with you the major features of translation. Okay. I have some more notes to give you here about this process, but any questions you'd like to ask? Okay, with the framework of the video in your mind, Kit, did you have a question? Kit, are you asking me why did the video say it made a protein when it was really a polypeptide? Is that what you're asking? Yeah, that's because you know, sometimes people kind of run loose and fast with terminology, right? Polypeptide and protein are like two sides of the same coin. It's the same coin, it's kind of the same thing, but as you get into more technical aspects of doing biology as a scientist, you do need to begin to appreciate that, well, there is a subtle difference here. Polypeptide is simply a chain of amino acids with no defined shape yet. When it folds, when you tie the bow, it now becomes a protein because now it will take on physiological activity. The video just was a little too simplistic to go to those higher standards, okay? Okay, other questions? Thank you, Yasmin. Yeah, absolutely, right? I mean, there's just no perfect scenario, right? It's kind of like no matter what you say, somewhere it's going to be confusing, okay? So that's why we're here to talk about this stuff. Okay. So let me see here. Yeah, so you know, as you are all pointing out, this link of colored balls on the screen is really simply a polypeptide. That's all it is at this moment. If it were to adopt a three-dimensional geometry, it would become a proper protein because then it would take on a physiological role in the cell. Okay. Okay. Anything else you'd like to ask? All right, let's keep rolling here. I'm going to keep rolling at least for a little while. Okay, so moving forward here, you should appreciate how the messenger RNA is read. Okay. How is the messenger RNA interpreted? That's sort of a question where you need a little bit of background, okay? The messenger RNA is scanned 3 nucleotides at a time by the ribosome. The ribosome scans the messenger RNA 3 nucleotides at a time, okay? A sequence of 3 nucleotides has a special name. It's called a codon. A sequence of 3 nucleotides is called a codon. And so hopefully from that you can infer that one codon specifies a single amino acid. Well, how do you know, or how does the ribosome know what these sequence of triplets, how does the ribosome know what these sequence of codons is specifying? Well, this refers to the genetic code. Okay, so I would ask you to look at figure 8.8. Take a look at figure 8.8 in your book. Figure 8.8 in your book. And please tell me, I'll just put this as a question in the chat window. No, in the notes. What does the codon, sequence ACA specify? You tell me, figure 8.8. What does the codon sequence ACA specify? Go ahead, type that into the chat window. What does ACA specify from figure 8.8? Okay, Does everybody see it there? Okay, Chelsea, thanks for the response. Yasmin, Rachel, fantastic. Morgan, good job. Okay, Ireland and Kit. All right, it does. It specifies THR, which is a type of amino acid. THR is an abbreviation for threonine. One of 20 amino acids. Okay, so the sequence of the codon, the triplet sequence of the codon, is interpreted by the machinery of the ribosome to tell the ribosome, okay, here's the next amino acid that I need to link into the polypeptide chain. It scans along the messenger RNA, 3 groups, 3 nucleotides at a time, 1 codon at a time, and it just keeps linking on the amino acids like a sort of like a ratchet motion, okay, as you kind of saw in the video. Okay, any questions? Okay, well, I'll put a couple questions in your notes. How about this? How does the ribosome know where to attach to the messenger RNA and begin translation? How does it do that? All right. Honestly, as I look across the information in your textbook, I'm not 100% confident your textbook tells you. Okay. What I'm going to tell you. Okay. Okay, Kid, I see your question. Just give me a second. So my question is, how does the ribosome know where to attach to the messenger RNA and begin translation? Here's the answer. The ribosome looks, and I don't mean it has eyeballs, because it doesn't have eyeballs, but it scans the RNA for a very precise RNA word, right? We've had DNA words so far in class today. Why can't we have RNA words? So the ribosome looks for an RNA word called the Shine-Dalgarno sequence. Okay, the ribosome looks for an RNA word called the Shine-Dalgarno sequence. When it finds that word, it will lock down and start assembling around the messenger RNA and begin its work from that point. Okay. All right. So, Kit, you have a question. Is there a difference between ACA threonine and ACU threonine? Kit, the amino acids that come out of those encodings are chemically identical. They are chemically identical. You know, there are some subtleties in terms of actually the transfer RNA molecules that bring the same amino acid. In other words, Kit, in your answer, your question, an E. coli cell might have 2 different types of transfer RNA molecules that bring the same amino acid threonine to the ribosome. And those 2 types are based on the different codon sequences that each respectively tell the cell to link in threonine next. So is there a chemical difference with threonine? No, but there will be a difference with the tRNAs that bring the same threonine to the ribosome. Okay. Okay. Other questions? I see your comment, Elena. It's okay. All right, so last thing here, and then we're going to move on. How is translation halted? You know, how does it stop? Well, I think you probably all know this. I bet you at some moment in your history, you've had to study some of this. And you know that there are these things called stop codons. Okay. Stop codons tell the ribosome to stop. Okay, you can see in figure 8.8 that there are 3 alternative stop codons that you might find in the RNA sequence that's being translated, any one of those will be interpreted by the ribosome as the endpoint where it should finish its work and not continue. Okay? So I know we've done a lot today. I still want to do more with you. Okay? Any questions right now? All right. So gang, I want you to think about these processes. I want you to learn and understand these new vocab terms, right? DNA words, RNA words, DNA helicase, origins of replication, single-strand binding proteins, RNA polymerase, replication fork, promoter, terminator. You see these, all these doggone words are going to jumble up if you don't be very careful with your study time. That being said, what I'm very interested in you obtaining some competency in and really understanding is being able to decode genetic information at a very basic level and getting correct answers out from it. So I'm going to give you a homework assignment, okay? And I'm going to put it into your notes here. So this is a homework assignment for next class. All right, I'm going to give you this. I'm going to give you a sequence of DNA, okay? And I want you, so let me write it out here. TAC, GGA, ATC, GTC, okay? I want you, through your homework, I want you to write out the RNA that would be made from this sequence. Okay, where's my, wow, I think my computer's going crazy here. Write out the RNA that would be made from this sequence. That's number one. And then from the RNA that you write, please write out the chain of amino acids that would be specified to the ribosome. Okay, so that's your homework. Given this starting DNA sequence, please write out the RNA sequence, and from the RNA that you develop, please write out the protein sequence, the polypeptide sequence, the chain of amino acids that will be made from it. Okay, let's see how you do with that, and we will talk about it next time. Okay, I'm going to go with lecture here, I would say for 15 minutes or so, and then we will stop and get to our lab work today. Okay, all right, so folks, at this point in chapter 8, we have talked a little bit about the structure of DNA. I have attempted to explain the basic idea and process of DNA replication. I have attempted to explain the major steps of gene expression, which are transcription and translation. Transcription, you make the RNA. Translation, you make the polypeptide, which many people just kind of sloppily say is protein. What we have not talked about yet, and which is so important, and which will end our time in chapter 8, I won't finish it today, I'll have to finish it next lecture period, is we need to talk about, well, okay, it's very nice that I now understand the basic idea of how a gene is expressed, but we have stated nothing about how does a cell know when to turn a gene on? And how does the cell know when to turn a gene off? In other words, it's nice to know how a gene is expressed, but how is it regulated? How is the process of gene expression regulated? And oh my, is that a loaded question, because we could take the rest of the semester on that topic, okay? It's still a topic that many scientists actively research, and we are very interested in the answers, okay? So we are going to shift gears from gene expression now to gene expression regulation. How do bacterial cells, okay, everything here is being explained from the bacterial point of view, because this is microbiology. How do bacterial cells know when to turn on the expression of a gene and when to turn it off? Okay, how's that happen? Well, the answer is we know some of the answer to that, but we don't know it all. We don't know it all. Okay. But your book has some examples of things. Okay. And so this topic is going to center around the idea of operons. Operons. Okay, let's talk about operons. Okay. All right, so I need to give you some background information here. Okay, and if you want some visualization, operons are talked about in figure 8.12, figure 8.13, figure 8.15. There's some nitty-gritty detail in there. Okay, so first off, operons are things that are primarily thought of as being prokaryotic things. We don't tend to talk about operons in the world of eukaryotic biology nearly as much. It's a little bit of a shame because there are some structures in eukaryotic DNA that are very much operon-like. But when you see operon in a textbook, that kind of means bacteria, okay? So operons are groups of genes. Operons are groups of genes that work together, and I'm going to put in parentheses, are expressed together to accomplish a common goal. Okay. For example, and this is the example that I'm really going to nail into you today and at least part of next class. Okay, an operon is a group of genes that are expressed together, that work together. The product of the genes being expressed works together to accomplish a common goal. Okay, one common goal would be how to metabolize and when to metabolize the milk, sugar, lactose. Okay. Now I want you to think about how important this topic is. Think about your favorite sports car. Maybe it's a Corvette, maybe it's a Lamborghini, maybe it's a Ferrari, maybe it's whatever. This is a kind of a silly analogy, but if I took all the parts in a disassembled manner for a Corvette and put them in a dump truck and drove the dump truck to your house and dumped all the parts to the Corvette in your front yard, you might say you have a Corvette. But philosophically, you really don't have a Corvette, do you? You have the parts that could be used to make a Corvette, but until you assemble them together in exactly the right sequence and the right relationships to each other, until you assemble them all together, you truly do not have a Corvette. And so the point of this conversation is cells have many genes. But if we don't know when to turn them on or when to turn them off, all you get is chaos in the cell. And in fact, that can happen and lead to cell death. So let me ask you this question in a similar vein in the chat window. Okay. What happens or what might happen if you apply your brake, I'm getting tired. I spelled that wrong. What happens if you apply your brake in your car at the wrong moment of time when approaching an intersection? What's going to happen, right? There's a couple possibilities here. Okay, you could crash, right? I mean, if you're halfway through the intersection before you apply the brake, somebody might sideswipe you, right? Or if you apply the brake completely before getting to the intersection, well, then you stop prematurely and you don't get anywhere. My point is, having a gene is like having the brake of your car. But you have to know when to apply it for it to do anything meaningful for you. You have to know when to turn a gene on and when to turn a gene off, or the gene doesn't do anything meaningful and actually can cause harm to the cell. So one common goal would be how to metabolize the milk sugar called lactose. Okay. Number 2. Okay. There are 2 primary types of operons. Okay. They're going to be operons that are called repressible operons. These operons contain genes which are normally turned on until they are intentionally turned off. They are normally turned on until they are intentionally turned off. Okay, so folks, in the chat window, tell me something in the chat window at your home. Tell me anything at your home that is normally on all the time until you turn it off. Tell me something that's on all the time until you turn it off. Okay, so televisions, computers, water, okay. What I was thinking of was the refrigerator, right? Isn't your refrigerator typically running all the time because you got to chill your food? And the only reasonable time you would turn it off is if you need to move the refrigerator or something like that, right? The refrigerator's on until you turn it off. That's kind of like a repressible operon. Okay. And so the other type of operon that exists is an inducible operon. An inducible operon contains genes that are normally off until you intentionally turn them on. Contains genes that are normally off until they are intentionally turned on. Tell me something at your home that's normally off until you intentionally turn it on. Okay, your washing machine, your shower. For many people, it's lights, right? The light switch is off until it gets dark at night, then you turn it on. This is the difference in operons that exist. Okay, so let me say this, okay? An example of a repressible operon, is the tryptophan operon of figure 813. Folks, I'm just here to tell you now, you are not responsible for understanding this because I have enough on your plate, okay? You are not responsible for understanding the trip operon. Let me highlight that in red. You are not responsible for that. If you want to go ahead and study it, go for it, but I'm not going to test you on it. However, I do have to test you on something. And so the other example of the operon is the lactose operon. That is the topic of figure 812. And the lactose operon, you are definitely responsible for. I will test you on some knowledge of the lactose operon, okay? Okay, let's continue here. I want to continue for 10 more minutes and then I'm going to stop. Number 3, some other things about operons, okay? There are times, such as in the repressible operons, where genes are normally on. They are just expressed all the time until you turn them off. Well, I'm going to tell you there's a category of genes that are always on in the cell and they never get turned off. They never get turned off. It's kind of like a leaky faucet that never gets repaired. Okay, so some genes are on all the time. They never get turned off. These genes are referred to as constitutive genes. Okay, these genes are referred to as constitutive genes. They are on all the time and they do not ever get turned off. Okay. All right. Any questions so far? All right. So what we need to do is get you started thinking about the lactose operon. And this is a conversation I will start today and I will finish it next week. And then we'll be on to chapter 13 and viruses. Isn't that exciting? Okay. So let's continue here. All right, let's draw a schematic of the lactose operon and neighboring segments of DNA. Okay. All right, so I think I can probably draw this faster and quicker with actual hand drawing. So let's do that. I'm going to draw a little schematic for you here.
Dr. Allen, would you mind scrolling back up just so I can see the last line for another second?
You bet.
Okay, thank you.
No problem. Okay. All right. Man, I'm tired. Let's keep going here. So let's write this map out. Okay, and here we go. All right. So I'm going to write a version of the lactose operon map that is similar to figure 812. It is similar to figure 812. I may go into a little bit more detail than that. Okay, so what we have here is P, I, CAP, P, O, Z, Y, A. Obviously, these letters mean something, and we're going to have to define them for you, and we will. But right now, we're just drawing a schematic. Okay. What you have right now is a schematic of some genes, and I'm going to define them, but it's also important that you know that what you see in front of you is simply a region. It is a physical section. It is a piece of the E. coli chromosome. In other words, this whole stretch right here is simply a region of the E. coli chromosome. Right? There are nucleotides of DNA present here in these symbolic representations of genes, right? This is just DNA. It's just a giant stretch of DNA that's part of the E. coli chromosome, and we're writing it out in a blueprint or schematic type way. Okay, so here's what I would begin to want you to appreciate. All right. So there are actually two operons here, right here and right here. This is the lactose operon. Okay. And there's a tiny operon to the left of it, which I'll put in orange, I guess. And that would be from here to here. And this would be another operon, and I'll blow it out this way. This is the lactose repressor operon. Okay. So the orange is something known as the lactose repressor operon. The green is something known as the lactose operon. The whole thing is simply a segment of nucleotides that are part of the E. coli chromosome. Okay. And so what we have to do next is define these things. Okay, so I'm going to do that. I'm going to give you the schematic legend. I'm going to give you the definitions. First off, let's talk about the lactose repressor operon. I know this is fairly heavy stuff, folks. It's not something you just absorb in one session, right? You're going to have to spend multiple study times thinking about this. Okay, so let me just number these things and say, okay, in the lactose operon, you see that P symbol. The P symbol is for the promoter. Okay, the promoter. Okay, this is as you recall from our previous conversation 20 minutes ago or whatever, the promoter is the binding site for RNA polymerase. Okay. To the right of the letter P, you have the letter I. The I is the gene symbol. It is a gene symbol. It's a letter. It's a representation of the gene called the lactose repressor gene. And what I'm saying to you is when I'm going to be very over the top with my next description because a lot of times things get said or written in books and you still can't follow because it's assumed that you understand certain things that maybe you don't understand, right? That's the hard part of education. The lactose repressor gene, okay? This is the DNA nucleotides, which represents the instructions to RNA polymerase to manufacture lac repressor messenger RNA. This is the DNA nucleotides, which represents the instructions to RNA polymerase to manufacture lac repressor messenger RNA. Once the lac repressor messenger RNA is made, ribosomes inside the cell will bind to the lac repressor messenger RNA and translate it into lac repressor protein. Okay, so we have here the lactose repressor gene. That's the instructions to make lac repressor messenger RNA through the process of transcription. When that messenger RNA is made in the cytoplasm of the cell, the ribosomes of the cell will sit down on the lac repressor messenger RNA and it will make lac repressor protein. Okay. Okay, I know I'm just about out of time here. Let's here's another you okay question. You okay? Anything you want to ask? Okay, another question. Type yes if you are passed out. Oh, come on. Somebody should type yes, right? That would be silly. Okay, all right. I'm about ready to pass out. Okay, here we go. Let's finish the legend and then we're going to stop. Okay, here we go. All right, so now we know what the lactose repressor operon is. Now let's talk about the lactose operon. Okay, first we have this cap thing. Okay, the cap thing is actually part of the promoter. This is a sequence that is a subregion of the promoter. It is a sequence that is a subregion of the promoter. Okay, cap is short for catabolite activating protein, which I know I haven't told you about yet. And what we have here in the operon, so cap is short for catabolite activating protein. Okay. Within the operon, cap is the nucleotides of the promoter that may, in some circumstances, be attached to by the cap protein. Okay, we need to be very careful here. What I'm saying is that there is a section of the DNA called cap, and I could even put this in parentheses, the cap binding site. Maybe that will help you, the cap binding site. Okay, cap is short for catabolite activating protein. There's also going to be a protein in the cell called the catabolite activating protein that under certain circumstances we haven't told you about yet will bind to this section of DNA called the cap binding site. Okay. All right. Number 2, P once again is the promoter. Okay. It is the promoter for genes Z, Y, and A, but I'm getting ahead of myself. Okay. So this is the promoter used by RNA polymerase to make when I say the promoter is used by RNA polymerase to make the proteins, what I'm saying is RNA polymerase makes the messenger RNA, and then the ribosomes decode the messenger RNA through translation to make the protein, right? So these genes are used to code for multiple sequences of molecules, with the final molecule being the protein. So P is the promoter is used by RNA polymerase to ultimately make proteins. We have this thing called O. Like, oh, holy crap, I have to know this kind of O. Okay, this is something called the operator. The operator sounds like a CIA hitman, doesn't it? But it's not that kind of operator. The operator is a DNA word. A DNA word that is the binding target of the lactose repressor protein. The story gets deeper, doesn't it? O is the DNA word that is the binding target of the lactose repressor protein. Z is a gene that ultimately codes for the enzyme called beta-galactosidase. Now we're cooking, aren't we? We're cooking now. All right. I told you the course just starts with chapter 8, okay? All right. So gene Z is a gene that ultimately codes for the enzyme beta-galactosidase. Okay? Beta-galactosidase is the enzyme that breaks down lactose sugar. Okay. Number 5, gene Y. Gene Y is a gene that ultimately codes for the enzyme permease. Okay. The enzyme permease. Permease is the enzyme that helps bring lactose into the cell. Permease, permeate, perforated notebook paper with holes. You see the connection there? Permease helps the sugar permeate into the cell. And then there's a gene A, which codes for an enzyme that is still a little bit of a mystery to scientists exactly what it does. Okay. What I can tell you about gene A is that this gene product has been studied for at least 50 years. And we're still a little bit confused about what it does. Hey, 50 years of effort, we can't figure it out. But it is what it is. There are some possibilities. This is a gene that ultimately codes for an enzyme that may help detoxify the inside of the cell from toxic waste products of lactose metabolism. This is a gene that we think it might detoxify the inside of a cell from toxic chemical waste products of lactose metabolism. From a biochemical perspective, this enzyme is known as a transacetylase, because it moves acetyl groups around from molecule to molecule. Who cares right now, right? You got enough on your plate. What you really are going to want to appreciate is definitely genes Z and Y. That's going to be important for our conversation. One piece of information up here I missed, and then we're stopping and switching gears. I didn't tell you that the lactose repressor gene, it is constitutive, meaning it is always on. It is always on. Okay. All right. Folks, that's a lot today. And we have maybe a half an hour of work next class to finish this up before we start chapter 13.
Can you move that up just real quick? Thank you.
Mm-hmm. Is there anything you'd like to ask before we switch gears? Because sometimes it's better to ask when this topic is fresh in your mind.
Thank you.
You're welcome. Okay, well, if you weren't passed out a little bit ago, you probably passed out now. All right, so let's stop this.